Welcome to the SANS Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump in, please take a moment to like, subscribe, and share this deep dive with friends and family. It really supports the SANS Science Experience and helps us keep unpacking these fascinating scientific breakthroughs for you. Today, we are undertaking a deep dive into, well, a really revolutionary paper. It kind of flips our understanding of exercise biochemistry on its head.
That's right. We're talking about lactate. You know, that molecule you probably associate with muscle burn during, say, heavy squats.
Exactly. And the mission today is to unpack this finding that something called lactylation of a protein, mTOR, is what actually signals your muscles to adapt during a workout. It's pretty wild.
It really is. So our goal is to get beyond just saying exercise is good and actually show you the molecular switch, how metabolic activity like making lactate links directly to cellular adaptation, specifically this recycling process.
And it uses lactate, which for so long was just seen as, well, waste.
Right. Dismissed as waste or maybe a simple fuel, this paper positions it completely differently.
So let's start there. The traditional view. It was pretty basic, wasn't it?
Very basic. Either it was a carbohydrate source, quick fuel, or, metabolic byproduct. Something linked to fatigue, something to get rid of.
But this new research says that's just wrong.
Well, maybe not wrong, but incomplete. Massively incomplete. The sources are clear. Lactate is a crucial signaling molecule. It's not just a consequence, it's a message.
A message that tells the muscle how to adapt after the workout.
Precisely. It mediates that skeletal muscle adaptation. It's a real paradigm shift, you know.
And the adaptation involves this process, autophagy, which is basically cellular housekeeping.
You got it. Autophagy is like the cell's cleanup crew, recycling old, damaged parts, making space for the new, essential stuff.
And exercise triggers this.
Oh yeah. Exercise is a huge activator for autophagy. It's vital for metabolic health. We even see it in mouse studies. If their autophagy is defective, their exercise capacity just tanks.
Okay, so exercise boosts autophagy and lactate is involved. How did they connect lactate as the trigger? Was there like a timing clue?
There was a really neat one, actually. They looked at mice doing a single bout of exercise, measured everything in the muscle, the gastrocnemius specifically.
The calf muscle, right.
And they saw lactate levels shoot up really fast, peak within about 10 minutes.
Okay, super quick.
But it's lactate.
Yeah.
The peak in autophagy, the actual recycling activity, that didn't happen until around the 30-minute mark.
Ah, so there's a delay. Lactate first, cleanup later.
Exactly.
Yeah.
That 20-minute gap strongly suggests lactate kicks things off. It's the upstream signal.
Okay, that's strong correlation, but correlation isn't causation. Did they prove lactate causes it? I think they used some special mice.
They did. The LDHA knockout mice. These mice lack a key enzyme, lactate dehydrogenase A, but only in their muscle cells.
Meaning they can't make lactate efficiently during exercise.
Right. And the results were, well, stark. These mice had much lower lactate, obviously, in muscle and blood. But they also had serious issues.
What kind of issues?
Their autophagy was impaired. Both baseline and exercise-induced autophagy were just lower. They couldn't clean up properly.
And functionally?
Functionally, it translated to smaller muscles, less lean body mass, and crucially, they couldn't exercise as well. Their maximal capacity was reduced.
Wow. So blocking the lactate signal really messes things up. It directly links lactate to autophagy and muscle function.
It's a very clear link. No lactate signal, no proper cleanup, poor performance and adaptation.
Okay, so lactate is the signal. But how does this small molecule, lactate, actually talk to the cell's machinery? This is where lactylation comes in, right? What is that?
This is the really groundbreaking part. Lactate signals through a new type of post-translational modification, a PTM. It's called lactylation, or CLA for short.
Like phosphorylation or acetylation, but with lactate.
Exactly like that. The lactate molecule itself, or rather a derivative, gets attached to proteins. It's a chemical tag. Think about it. The cell is using a direct readout of its metabolic state, lactate, to chemically modify its own proteins.
That's incredible. Using its own energy currency as an instruction label.
Right. We knew it happened on histones, affecting genes, but this paper shows it hitting other key proteins directly involved in cell processes.
And the main target here is a big one, mTOR complex 1.
The absolute master regulator, mTORC1.
Yeah, and mTORC1 is usually involved in growth, right? Kind of puts the brakes on autophagy.
Exactly. mTORC1 is a go signal for growth. Building proteins, getting bigger. It actively suppresses autophagy. So if you want a clean house, which exercise adaptation requires, you need to turn mTORC1 off.
So the lactate signal, this lactylation, needs to hit mTORC1 and switch it off.
That's precisely what the paper shows. Lactate ramps up autophagy by inactivating mTORC1, and the key discovery is where this happens.
Okay.
Lactate promotes the lactylation of the mTOR protein itself, right at a specific spot, lysine 921, K921.
So putting a lactate tag on K921 is the off switch.
It acts like that molecular switch, yes, to suppress mTORC1 activity. They showed it in muscle cells. Treat them with lactate, you get more mTOR lactylation and less mTORC1 activity. You see less phosphorylation of its downstream targets like ULK1.
Okay, but how does just tagging that one lysine residue shut down the whole complex? What's the mechanism?
Well, the tag seems to change the complex's shape or accessibility. The lactylation at K921 allows other proteins, negative regulators, to bind more easily.
Like molecular brakes.
Exactly. Specifically, proteins called TSE1 and PER2. The paper shows K921 lactylation enhances their recruitment to the mTOR complex. So the tag acts like a docking site for the stop crew.
Lactate tags mTOR, the brakes jump on, and protein building stops, allowing cleanup to start. That's really elegant.
It is quite elegant, yeah.
But science always needs that final proof, right?
Yeah.
If K921 lactylation is the switch, what happens if you break it? If you can't lactylate that spot, even with high lactate?
Right. And that's exactly what they tested with the mTOR K920My. They mutated that specific lysine K to an arginine, R, at position 921. Arginine can't be lactylated.
So these mice make lactate, but the off-switch receptor on mTOR is broken?
Precisely. The muscle can't read the lactate signal correctly at that crucial point.
And the result? Did it confirm the hypothesis?
It did, quite dramatically. These K921R mice showed sustained mTORC1 activation. The off-switch was jammed on.
So no suppression?
No suppression. And consequently, they had impaired autophagy, just like the low-lactate mice. Muscle function was reduced, muscles were smaller, and exercise capacity was down.
Wow. They look just like the mice that couldn't make lactate in the first place.
Functionally, yes.
Yeah.
It really pins down the necessity of that specific K921 lactylation event. Making the signal isn't enough. You have to receive it correctly.
And there was another layer to this, wasn't there? Something about how this mutation affected other signaling pathways.
Right.
Like AMPK.
Yes, this was really interesting. AMPK, the AMP-activated protein kinase, is another major energy sensor. Usually during exercise, when energy is low, AMPK helps shut down mTORC1.
Right, another brake system.
But in these K921R mice, mTORC1 became resistant to being shut down by AMPK.
So the lactylation wasn't just one way to turn off mTORC1. It was somehow required for other pathways, like AMPK, to do their job properly, too.
That's what the data suggests, that K921 lactylation is needed for the full engagement of AMPK's regulation of mTORC1 during exercise. It's like the lactate signal primes mTORC1 to listen to other stop signals like AMPK.
Okay, that makes this even more central. It's not just one isolated switch. It's integrated into the whole energy-sensing network of the cell.
Absolutely. It provides this beautiful conceptual framework for how muscle translates metabolic state lactate levels directly into adaptive responses like autophagy, coordinating with other pathways.
Which obviously has broader significance. You mentioned therapeutic potential.
Yeah, there's definitely potential. If you could modulate this mTOR K921 lactylation, maybe you could help treat conditions where muscle autophagy is messed up. Things like muscle wasting, atrophy, maybe even some metabolic disorders.
Restore that healthy cleanup process.
That's the idea. Restore healthy autophagic flux.
But the paper urged caution too, right? It's not straightforward.
No, definitely not. Biology rarely is.
Yeah.
The big challenges are specificity and dosage. You need to target muscle tissue specifically. You don't want to mess with mTOR signaling in the brain or liver accidentally.
Right.
And you need the right amount of change. Too little autophagy is bad, but too much autophagy can also damage muscle. Finding that therapeutic window, that sweet spot, is really tricky.
So a promising target, but needs very careful, precise approaches.
Exactly. It's complex biological engineering needed down the line.
Okay, so let's wrap up this deep dive. The big picture is lactate is not just metabolic noise or simple fuel anymore. It's a critical signal.
A vital chemical command.
It uses this novel modification, lactylation, specifically at lysine 921 on the mTOR protein.
Right, that's the switch.
To turn off the growth-promoting mTOR complex 1, and turning that off is key because it unleashes autophagy, the cellular cleanup crew.
Which is absolutely essential for muscle adaptation, getting stronger, and staying healthy after exercise.
It really does redefine a fundamental piece of exercise biochemistry.
It absolutely does. So The key takeaway for you listening is this. Next time you're working out hard and you feel that burn, then
Yep.
lactate isn't just waste.
It's a signal.
It's your muscles sending the command. Time to clean up and get stronger.
An excellent thought to leave you with. Thank you for joining us for this analysis of cutting-edge molecular biology. We hope you found this as fascinating as we did. Remember to like and subscribe to the SANS Science Experience for more deep dives into cutting-edge science. We'll be back soon with more insights from the world of preprints and papers.